Welcome to microbiology. We are going to be starting off with chapter 1, which is a little bit of everything microbiology. And this is going to set us up into some of the topics that we will be talking about this semester. So one of the things that happens in micro that I don't really mean to happen is that I make some germophobes. Not that I really want to make germophobes, but I think it's more of a general awareness of what is actually on us, in us, and around us. So microorganisms are everywhere. In fact, there are so many microorganisms on us and in us, they actually outnumber our own cells. The good news is, even though we know that many of these microorganisms cause disease, very few of them are actually pathogenic that actually cause disease. We are going to be talking about some of those organisms that are pathogenic, and we'll talk a little bit about what pathogenic means as well as what virulence means. But the good news is that a lot of that bacteria does a lot of good for us. They are essential to our food web. We see them in a variety of industries. They help us make vaccines and medicine. They can even help us to manufacture things like beer and wine. All you have to say is beer and wine, and I'm sold on microbiology. So this is a little bit of the cast of characters that we will be talking about. The majority of our time will be spent talking about prokaryotic cells. They are different from eukaryotic cells, and we will go through some differences and similarities between prokaryotic and eukaryotic cells. So we'll do a little review of general biology. Bacteria fall under the group prokaryotic, meaning that they don't have a nucleus. They also don't have a lot of those fancy cell organelles, so those membrane-bound organelles like mitochondria, endoplasmic reticulum. We are going to be spending a good portion of the semester talking about bacteria, characterizing bacteria, and looking at them underneath the microscope. These organisms are very tiny. We are not going to be able to see them without a microscope. So we are also going to be spending quite a bit of time using the microscopes, learning how to focus under oil immersion. I always say to students that if you were that student in previous classes that when your lab partner focused beautifully under the microscope and you were applauding them, I always say that you're going to be applauding yourself because we're all going to get proficient at using the microscope and focusing under oil immersion. Some other characters that we will be looking at this semester are eukaryotic cells. So things like fungi, protozoan. So if you ever looked at pond water underneath a microscope, you most likely were seeing a lot of those protozoans. We will be talking about protozoans in the parasitology chapter. We'll also be talking about some bigger things like helminths, like worms, in the parasitology chapter. This is also what I term the heebie-jeebie chapter. We'll kind of be itching and scratching our way through that. It happens to be one of my favorite chapters. And we will be talking about some of those things that go bump in the night. We'll also talk about some acellular things as well. So things like viruses and prions. Viruses are not considered living things. They're not cells. They have a nucleic acid core. So it's either RNA or DNA, and that is protected by a protein coat. So we're going to talk about some of the different viral diseases and how viruses replicate, but they are acellular. They are not considered living things because they cannot undergo all of those characteristics of living things that we talk about, such as metabolism, generative functions, and maintaining homeostasis. So microbiology really varies, and we can find it in a lot of fields. I know many of you are probably in healthcare or going into health sciences. So you're interested in microbiology from a clinical perspective. So what are those organisms that are causing disease? How can you help patients deal with those different infections? That certainly is part of microbiology, as well as studying how microbes pass through populations. So looking at epidemiology. At the end of the semester, we'll talk about epidemiology, why it's important to know how things cause disease so that we can prevent outbreaks. And some of our last chapters that we will talk about is on immunology. And this is how our body can fight these pathogens. If you name an industry, I'm pretty sure that we could find some sort of microbiology applications. My thesis was really in environmental micro, and I will be talking quite a bit about that this semester, especially in the first few chapters. So the bacteria that I studied and characterized were obtained off of these tree needles, Tamarax aphylla, which is kind of like a pine tree. And I characterized those bacteria. They came from a desert, so they had some unique living requirements that I'll talk a little bit about. We will mostly be focused on clinical microbiology, so some of those pathogenic organisms. We'll also talk about some industrial applications as well. So we can see microbes and microbiology in a variety of industries and fields. All right, so microbiology, even though they had no idea that these tiny organisms actually existed, really date back very, very long ago. I think my first encounter with microbiology was learning about the Black Plague or the bubonic plague, which is caused by Yersinia pestis. And Yersinia pestis was a bacteria. It was carried on fleas, and those fleas were on rats, and it killed millions of people. So I remember learning about this as a child. If you ever played the game Ring Around the Rosie, that's actually about the bubonic plague. So I remember doing this in school and learning about what that song was actually about. So I think that was my first introduction into microbiology, hearing that something so small killed millions of people. I'm not going to lie, it was pretty alarming. And so that really was the first time that I started to learn about microbes. Like I said before, not all of those microbes are going to be pathogenic. And some of them are, and we will talk about those. But I remember very clearly learning about this and being quite alarmed at what I was learning. So as I mentioned, we are not going to be able to study these microbes unless we look at them underneath the microscope. So we're going to talk a little bit about some of the history and some of the scientists that first looked at living organisms underneath the microscope. And before they had the invention of the microscope, they really had no idea that these tiny organisms actually existed. One of my favorite things to do is to look underneath the microscope, and I always get a kick when I'm teaching students, especially in Bio 109, if they haven't used a microscope before, how to focus the microscope and then look at pond water underneath the microscope. And I get such a kick out of students that see this whole new world open up when they see all of those living organisms underneath the microscope. I can't even imagine what it was like to actually be one of the first scientists to see this microcosm open up. So the two scientists that we mostly talk about with microscopes, Robert Hooke. So he was the first one to look at non-living things underneath the microscope. In fact, he used the first compound microscope. In fact, we will be using a compound microscope. That basically means that it has two lenses. And he observed cork underneath the microscope, and he noticed that within the cork there were these little pores, these spaces, and he named those spaces cells. And that's actually what we use today to term cells, the basic structure and function of living things. Anton von Leeuwenhoek, he was the first to observe living organisms underneath the microscope. So he was able to see those pond swimming things. I can't even imagine what that was like to actually see this whole new world underneath the microscope open up. So the microscope that he was using could see up to a magnification of 300 times. To give you a little idea of how strong that is, our compound light microscopes, our high power will go up to 400 times magnification, which gives us some good visuals of certainly eukaryotic cells, but we can even make out some of those prokaryotic cells. So he was actually using a very strong microscope. We're going to be spending quite a bit of time, especially the first part of the semester, looking at organisms underneath the microscope. We're going to have lots of practice focusing the microscope, and pretty soon you'll be applauding yourself for being able to focus under oil immersion. All right, so unfortunately, some of these microorganisms do cause disease. So the germ theory of disease is that microorganisms can invade other organisms and cause disease. So this was kind of earth-shattering news years and years and years ago, way beyond before I existed, believe it or not. And the reason why this was earth-shattering news was that many people believed in spontaneous generation. And they believed that you could actually get microbes and living things from non-living matter. Now, this might seem kind of ridiculous because, of course, we know the cell theory of disease. The cell theory is that all living things come from other cells. But at the time, if you think about before they had refrigeration, before they had modern amenities, even screens on windows, they had a lot of things happening in kitchens in their house. And so what they found was that if they had some meat on a counter, and they knew that meat was non-living because they had killed the animal, that they found that there were these little tiny wormies that were actually kind of crawling on this meat. So they thought, well, apparently you can get living things from non-living things. So there was a scientist that did not really buy into this spontaneous generation. And this scientist was Francisco Redi. And so what he did was he set up an experiment. So he set up this bell jar experiment where he put 3 pieces of meat into each of these bell jars. And in one jar, he left it completely open. The other jar he closed off. And then the other jar, he put some cheesecloth over the top. And cheesecloth is kind of like gauze. And what he noticed in the open jar, these flies were actually coming in and they were landing on the meat. Before long, he noticed that there were these little wormies, the little maggots that were actually crawling on the meat. So he realized based on this experiment that it wasn't the meat giving rise to living things. It was actually the flies that were coming in that were living, laying their eggs, and the larval form were actually those worms that they saw growing on the meat. So he started to do some work in spontaneous generation to disprove this. So there were some other scientists that wanted to continue to do some work with spontaneous generation. Some of them weren't completely buying into Francisco Redi's work. Spallanzani, what he did was he boiled and sealed liquids to show that they would not grow microbes. Needham was still kind of holding on to the spontaneous generation theory. And he said, well, Spallanzani, the reason that you're not seeing anything growing is that there's no air that's coming in, and the air has the living things. So Pasteur, and let me move my little screen down here so that we can actually see Pasteur's setup. Louis Pasteur, and we'll talk a little bit more about him. He used these swan-necked flasks. So he said, okay, let's boil the liquids, but let's add this little glass tubing so the air can actually get in there. And because the microbes ended up getting stuck in that curve of the glass, no microbes actually grew. So he was also trying to disprove that spontaneous generation. We usually think of Pasteur with pasteurization. So this is heating things up. We usually think about this with milk. It's not necessarily sterilizing, but it's just heating it up enough to reduce that microbial load, to reduce the number of bacteria. Pasteur also worked in immunology, and he actually helped to start the rabies vaccine. So we'll see Pasteur a couple of times throughout this semester. So one of the things that we will do this semester is to grow pure cultures. And a pure culture means that we have captured only one organism. And when they first started trying to grow microorganisms in the lab, these microorganisms, because they liked the human body environment, they had to be incubated at 37 degrees Celsius, which is body temperature, 98.6 degrees Fahrenheit. But the problem was trying to actually grow these organisms on a solid surface. And so they were trying to use gelatin. But the problem with gelatin, and if you think about Jell-O, when you have to refrigerate it for it to congeal, if it's kept at a warmer temperature, especially 37 degrees, it was melting. It wasn't really holding up its consistency. So one of the assistant's wives suggested, well, we use this agar, or if you prefer to pronounce it agar, to thicken things in the kitchen. And so they actually tried this in the medium and it worked. And so they were actually able to grow organisms on a solid surface. This is actually what we use today. So this is the thickening agent, which is in our media that we use to pour our plates so that our organisms can grow on a solid surface. When we grow them on a solid surface, we're able to see them. And so we can see if there's contamination or if there is one culture. So Robert Koch was doing some work with maintaining pure cultures. And he was also doing some work to prove which organisms caused which disease. And so he came up with this series of tests. And so I have a little picture here where you can see how he conducted his tests. So first off, he said that this microorganism must be found in all organisms suffering from the disease. So what he did was that he was able to remove that organism from individuals that were inflicted with that disease. The second part of his postulate said microorganisms must be isolated and grown in pure culture. So once they were removed from the organism, then they had to be grown in pure culture. Then he said, once we grow it in a pure culture, then we can take it, put it into a healthy organism, and it should cause the same disease. He said, finally, in that organism, we must be able to re-isolate it and then grow it on a pure culture. So going through these series of tests, the postulates allow to prove which organisms caused which disease. And so we call those Koch's postulates. So we are going to be working to maintain pure cultures this semester. The picture that you are looking at is an isolation streak. And as you can see, this organism has sort of an orange color. You notice there are no yellow organisms or creamy white organisms. So this is a pure culture. So we work really hard in the lab to maintain our pure cultures, especially if we're using them semester after semester. So it's really important that we don't get that contamination. So many of the things, the techniques that we will be using this semester is going to be to maintain those pure cultures. And so these are some of the techniques that we will be learning about this semester. So now that we know that these organisms can cause disease, how can we actually prevent disease? So Semmelweis actually worked to improve hygiene and sanitation and had the gall to suggest to doctors, hey, maybe you want to wash your hands after performing an autopsy before you go in to see a live patient because you are actually spreading disease. Can you imagine suggesting that to a doctor? I mean, everything that we know about disease and how hand washing is our best prevention, the doctors were actually kind of disgusted by Semmelweis's suggestion. Florence Nightingale, who revolutionized nursing, actually helped to improve hand hygiene, even though Florence Nightingale wasn't completely convinced about microorganisms, although she was later on in life. She greatly reduced the number of patients that died because of her hygiene practices. Another scientist, Lister, and what does Lister sound like? Sounds kind of like Listerine, right? So when we think about Listerine, when we're trying to kill the germs in our mouth, Lister actually helped to disinfect some things during surgeries. And so what Lister used was this chemical, carbolic acid, which is kind of like the gold bar standard of disinfectants that we'll talk about this semester. He used that and helped to develop the first aseptic technique. So one of the things that we will be learning about and practicing this semester is aseptic technique and perfecting our aseptic technique. So I like to think of aseptic technique as sort of a 3-part technique. So first of all, this is not contaminating our surroundings. It's not contaminating ourselves from the cultures that we are working with. And it's not contaminating our pure culture. If you work in healthcare, chances are you are already using aseptic technique. So when you are working with a patient, if they have a disease that is pathogenic that could be carried and disseminated to other people, you certainly want to protect yourself from getting sick. You want to protect your surroundings. And of course, you don't want to introduce new disease into your patient. So all of those gowning, degowning, and all of those techniques that you use to keep yourself and your patient safe are aseptic technique. We're going to be using it on a smaller scale when we work in the lab and we work with our pure cultures. So this is something that we are going to be spending quite a bit of time perfecting over the semester. All right, some other topics that we will be talking about this semester. As I mentioned earlier, later in the semester, we will talk about immunology. And immunology is how our immune system interacts with pathogens. So how do we protect ourselves from getting disease? So those are going to be our 3 final chapters. Immunology is one of those topics in science that they are still learning about. We're still studying. So it's kind of one of those last uncharted territories. I mentioned viruses earlier. We are going to be talking about viruses, so some different viral diseases, and also how viruses replicate. We don't usually say reproduce with viruses, because viruses are not considered living things. So they would not undergo those generative properties. So viruses are intracellular obligate parasites, and they cannot replicate without a host cell. Some other things that we will be talking about are chemotherapies. And usually when we think of chemotherapy, we think of things really strong to treat cancer, where you have to go into a hospital to get an infusion. Chemotherapy is basically using any chemical substance to treat a disease. We'll also be talking a little bit about some of the cool things that bacteria do with genetics and molecular biology. So we will talk about DNA. We'll also talk about how we can use some of those cool things that bacteria do to help us to make medicines and vaccines. So that's going to be coming up soon in one of those first chapters. We'll also be talking about vaccinations, especially when we get into the immunology chapters. Vaccine is actually derived from the word cow, which is vaccinia, and this is based on Edward Jenner's work. And Edward Jenner was doing some research on smallpox, and smallpox was a deadly disease that we have eradicated, fortunately. And when he was learning about smallpox, he was also researching a very closely related disease, which was cowpox. And so he noticed that the milkmaids that were milking the cows would get cowpox. However, they were less likely to get smallpox. So he said, well, perhaps that people that are exposed to cowpox, they're actually less likely to get smallpox. So what is it about the cowpox that's allowing people to evade getting sick by smallpox? So he did an experiment, a very unethical experiment, at least in today's standard. And what he did was he took fluid from a cowpox blister and gave it to a small boy. Then he infected the boy with smallpox. Now, smallpox was fatal. And fortunately for Jenner and the boy remained healthy. So there was something about that cowpox that allowed him to be immune to that smallpox. Hence, immunology was born and vaccinations were born as well. I mentioned that Pasteur also did some work with developing the rabies vaccine as well as cholera vaccines. And what he was doing was he was working with chickens and cholera. And when he went to infect the chickens with cholera, he accidentally used an old bacterial stock. So upon realizing his mistake, he decided, okay, well, let me take that new live bacterial culture, the new one, the fresh one, and inject it into the chickens. Well, what he found, the chickens stayed healthy. So again, here is that question of why did the chickens stay healthy? There must have been something about that old bacteria. It was enough not to make them sick. However, it allowed them to remain healthy when that new live bacterial culture was injected. So again, here is that immunity protecting those chickens. Jonas Salk is somebody else that we also think of with vaccines. And Jonas Salk helped develop the first polio vaccine. Polio is one of those things that we had hoped to eradicate. Unfortunately, we are seeing more cases today. In the past year or two, I know in Rockland County, New York, they were testing some wastewater, and they actually found some polio in the wastewater. So were people asymptomatic? Were people having actual infections? And one of the things that we will talk about with vaccinations is people choosing not to vaccinate. And this is unfortunately allowing some of these diseases that we haven't seen or had to worry about in years actually becoming real problems. So we'll talk about vaccines, why some people choose not to vaccinate. There are some reasons that people cannot get vaccinated. So we'll talk a little bit more about the different types of vaccines and how they work. I mentioned viruses, so we are going to be spending a little bit of time working with viruses, or at least talking about viruses. We are not going to be able to see any viruses in lab. because it would take a very powerful microscope, an electron microscope, to actually see them because they are so small. And again, microbes were very small, and until the invention of the microscope, they had no idea what was causing diseases. Well, once they discovered microbes, guess what? There was something even smaller that was causing disease. So there was a scientist, Chamberlain, and he was actually filtering some bacteria and discovered that even after he had filtered the bacteria, there was something else even smaller than bacteria that was actually able to cause infections. Well, lo and behold, they were actually viruses. And what I have here are some different structures of viruses. So we will talk about some viral diseases, their structure. The picture next to it is actually the bacteriophage replication cycle. And so bacteriophages, these are viruses that only affect bacteria. So we'll go into a little bit of their replication and replication cycle. And then we know that we need medicines to actually treat these diseases. Some of our first medicines were actually plant-derived, and in fact, we still use some of those medicines today. So, for example, the bark of the cinchona tree was used to treat malaria. Today, we know this to be quinine and derivatives, and this is something that we actually still use today. Alexander Fleming was kind of noted as the father of antibiotics. If you were to do a Google search, you would probably find some other scientists that would also be listed as some of the first people to work with antibiotics. And Alexander Fleming, he was working with bacteria, and he noticed that lysozyme within saliva actually killed bacteria. Fleming also noticed that when he was growing bacterial cultures, that there was something on his plate that was a contaminated plate was actually killing the bacteria. He observed that what was killing the bacteria was a fungus that was growing on the plate that the plate was contaminated with. So that fungus was actually releasing a substance that was killing the bacteria. In fact, that is the true definition of an antibiotic, that it is actually released by another living organism. We tend to use antibiotic and antimicrobial therapy interchangeably. One of the things that we'll be talking about this semester are different antibiotics or antimicrobial therapy, how they work on bacteria, how they don't affect our cells. And we'll also talk a little bit about resistance, because we know that resistance is a huge issue with treating diseases. And then finally, we'll be talking about some cool things that bacteria do. So bacteria can actually transfer genes to one another within the same culture. This is what we call horizontal or lateral gene transfer. So finally, we will be talking about some of the cool things that bacteria do. So they can actually undergo horizontal or lateral gene transfer. So they can actually transfer genes within the same generation. This is something that eukaryotic cells cannot do. And of course, this compounds that problem of resistance. So we'll be talking about some of the unique things that bacteria do, as well as how we can actually take advantage of that in the lab and help us to make vaccines and medicines that we can use. So the one thing that I love about microbiology is that it is so interesting and so varied. So we will be hitting quite a few different topics. The other thing that I love about micro is the lab. So some of the things that we will be doing in lab, they do in labs all over the world. So once you finish microbiology, you could actually get a job as a microbiologist in a lab because of the techniques that we learn. So I hope that you enjoy microbiology, the varied topics, and I hope you enjoy working in lab as well.